Hello everyone, welcome back to my YouTube channel. So today's video it's all about the nutrition in bacteria. So bacteria exhibit different types of nutrition classified into autotrophic and heterotrophic bacteria. Autotrophic bacteria, first we discuss the or learn the autotrophic bacteria. These bacteria can synthesize their own organic food from inorganic raw material. Okay. So synthesize their own organic food from inorganic raw materials. They use carbon dioxide as a carbon source. They can be further divided into photoautotrophs bacteria and chemoautotrophic bacteria. Now first, photoautotrophic bacteria. So possess the photosynthetic pigments like bacteriochlorophyll or bacterioviridin in their membrane-bound lamella. Okay, in this picture you can see this is the DNA. This is cyanobacterial cell, suppose. Okay, for example, for instance, we can say the cyanobacterial cells, thylakoid and lamella. Okay, which having the bacteriochlorophyll and bacterioviridin. Okay, they carry out photosynthesis but differ from the plants. Water is not used as electron donor. Instead, they use hydrogen sulfide, another ammonia, hydrogen gas as an electron donor. Oxygen is not released as a byproduct. In photosynthesis, oxygen is released as a byproduct, but here it's not. Now the example reaction, CO2, hydrogen sulfide, light, and it's produces sugar, sulfur and water. So example of the bacterial cells, so green sulfur bacteria, chlorobium. Another one, purple sulfur bacteria, chromatin. Now, I here mention some of the points, like there are two main types of photoautotrophs. Oxygenic photoautotrophs like cyanobacteria plants, as we know, and they use water as the electron donor and release oxygen as a byproduct. Okay. And second one, it is the anoxenic. Okay, anoxygenic photoautotrophs like purple sulfur bacteria, here mentioned here in slide, and green sulfur bacteria. So they use the other substances, okay, like as I mentioned, like hydrogen sulfide is the electron donor, and oxygen do not produce the oxygen as a byproduct. Now come to the second type of autotrophs, autotrophic bacteria. One is the autotrophic bacteria. Okay, now second one is the chemoautotrophic bacteria. So synthesized organic compound. Okay, see, organic compound, lipid, protein, anything. Using chemical energy, where derive the chemical energy or obtained from the oxidation of inorganic substances. Okay, so inorganic substances may be ammonia, sulfur, okay, hydrogen sulfide, okay, anything else, maybe inorganic substances. They do not require sunlight, yes, okay. Uh, Autotrophs, okay, here it is in the next previous slide, autotrophic bacteria. First, a phototrophic, they require the light, but here it's not required the sunlight. Okay, so types of chemoautotrophic bacteria. One is nitrifying bacteria. Okay, oxidized nitrogenous compound. For example, we understand the reaction like here oxidation of ammonium ion NH4 plus into nitrate Na2 by chemoautotrophic bacteria, specifically nitrifying bacteria like nitrosomonas. Okay, it is reaction happens when the nitrosomonas bacteria, okay, through the nitrosomonas bacteria. Example of bacteria, this is structure of nitrosomonas, and second is nitrobacter. Okay. Second type of chemoautotrophic bacteria, that is sulfur bacteria or oxidized sulfur compound. Okay. Example reaction, we understand it deeply, like hydrogen sulfide. Okay. Reduced in this reduced sulfur compound often found in volcanic region. Okay, they found in deep sea, also deep sea vents and decaying organic matter. Act as the electron acceptor. Okay, H2S is act as the electron acceptor. Now product as sulfur. Okay, here it is. This is a reactant and this is the product, entire product. Elemental sulfur deposited either outside or inside the bacterial cell. Okay. And second, H2O form as a result of the oxidation process. Energy released and used by the bacteria for biosynthesis, okay, to make food from CO2. Example of sulfur bacteria, Tharabacillus, and second is Begeto. Yeah, Begiato. You can pronounce this as both way. Now, third, iron bacteria, oxidized iron salt. So reaction, we see the reaction, how they work on this iron salt. Okay, so first reactants, FeCO3, here is the FeCO3, iron carbonate. This is the source of ferrous iron Fe plus 2, right? And act as the oxidizing agent. Okay, here is O2 and it act as O2 act as the oxidizing agent. H2O needed for the chemical conversion. Now product, FeOH3, okay, ferric hydroxide, an insoluble or rust color precipitate often seen in iron rich environment. Okay, when you see in iron rust, okay, CO2 released from the carbonate group. Okay, this one. Energy generated and used by bacteria to synthesize their own organic food from CO2. Now the example of the bacteria, Acidothabacillus ferroxidase. Okay, now second type of bacteria, heterotrophic bacteria. Okay, earlier I mentioned the phototrophic bacteria, autotrophs. Okay, autotrophic bacteria, and second part is the heterotrophic bacteria. So heterotrophic bacteria divided into three: saprophytic bacteria, symbiotic bacteria, and parasitic bacteria. Okay. So heterotrophic bacteria cannot synthesize their own food, dependent on external organic sources. Okay, so divided into three: saprophytic bacteria, decomposer, is we can say also the decomposer, symbiotic bacteria, mutual beneficiary, parasitic bacteria. Now first, saprophytic bacteria obtain food from dead or decaying matter. They break down complex organic materials into the simple soluble form. The example of Bacillus subtilis. Okay, this is a microscopic structure of Bacillus organism. Okay, second, symbiotic bacteria, mutual beneficiary. Okay, live in close associated with other organism and benefiting both. Example, Rhizobium, fixing nitrogen in leguminous plant roots. Escherichia coli helps in vitamin K production in the human intestine. So this is the nodule of leguminous plant. In these nodules contain the rhizobium. Okay, nitrogen fixing bacteria. Okay, this is structure of microscopic view of rhizobium, and this is the Escherichia coli structure. Third, that is the parasitic bacteria pathogens. Derive nutrition from living organism, host, anything, human, animal, anything. Okay, often causing disease. They mostly cause the disease of the host, in the host, right? Okay. Example, Mycobacterium tuberculosis cause tuberculosis. Second, Treponema pallidum. Okay, this bacteria present in mouth, human mouth. so cause the syphilis. This is the structure of Mycobacterium tuberculosis. Second one, this is a Treponema pallidum. Okay, spiral shape. Okay, now come to the another topic, that is differences between exotoxin and endotoxin. Okay, so bacteria produce the toxin that can harm host organism. Okay, that toxin can be classified into exotoxin and endotoxin based on nature and mode of action. So features like produced by who is produce the exotoxin and endotoxin. Gram positive always produce the endotoxin, exotoxin. Gram negative endotoxin. Also known as a protein toxin, and here is a lipopolysaccharide toxin. Released from actively secreted outside the bacterial cell. Exo, that means the outer side of the cell. Endo means inner side, okay, inside of the cell. Release when the bacterial cell lies, okay, when bacterial cell is dead, then and then only release this endotoxin, okay. Exotoxin when released where the bacterial cell must be live. Heat stability, heat liable, easily destroyed by heat, okay, 60 to 80 temperature you can destroy this toxin, bacterial toxin. Heat stable, resistance to heat. Toxicity, high toxic even in small amount, okay, 0.00 microgram even, okay, they highly toxic. Less toxic, require large amount of host damage. Okay. So this is the all about today's video. I hope you enjoy it. In the next lecture, we will be discussed about the respiration and reproduction in bacterial cells. So thank you so much for watching this video, and don't forget to subscribe my YouTube channel.